Today we are going to discuss the paper number 1, 9700, variant 1 to October November 2023 from question number 1 to question number 20. Now I would like to move towards the first question, and first question is the which combination of lenses for a light microscope will give the greatest magnification? And students, you know the formula of magnification. And here we are going to find out which combination of lenses for a light microscope can be. So here I would like to 100 into 5 is 500. 40 into 10 is 400, and 40 into 15 is 600, and 15 into 100 is 1500. So according to this, the greatest magnification can only be the D. So here in this case, the first question, the D is the correct option. Now question number 2. The diagram shows an electron microscope of virus particles in human nucleus. This is the virus particle, and its emitter can, if we measure it, then it can be around 3.5 or 3.6, between 3.5 or 3.6, and millimeter. It is in millimeter. We can change it into, first of all, into micrometer. So 3.5 multiplied by 1000, so it will be 3600 micrometer. Then we will change into micrometer to nanometer. 1, 2, 3. So it will be in nanometer. And what will happen? And when we apply this formula, then we can have to image over magnification. So it will be divided by 24,000 by magnification. So the answer will be 150, or when we, it will be the correct option over here. That is, if we divide this, you can see, students, you can do it by yourself. Then you can divide by using calculator. You can find out the correct option. So this is the correct option, which is 1.5 into 10 raised to power 2 nanometer. Now, question number 3. Question number 3 is, let me do it. Question number 3 is, what is correct about the synthesis and release of the glycoprotein mucin in goblet cells? You know about goblet cells, which produce the glycoprotein mucin. The protein is produced on ribosomes, and a carbohydrate chain is added in the Golgi body. Yes, the glycoproteins are packed into vesicles in the Golgi body, forming lysosomes. No, lysosomes are hydrolytic enzymes. So according to this, always it is wrong, because lysosomes produce the hydrolytic enzymes. Then third is the secretory vesicles containing the glycoprotein move from the Golgi body and fuse with the cell surface membrane. So it is also the correct definition or statement. So according to this, the B is the correct option in this case. Now question number 4. Question number 4 is, what are functions of microtubules? What are microtubules? You can say they make the cytoskeleton of the cell. So they have given us the three statements: allowing movement of cilia in a bronchus, yes. Attachment to centromeres during metaphase, yes. Moving secretory vesicles around a cell, yes. They perform all these functions. So according to this, A is the correct option. Which organelles, question number 5, which organelles contain nucleic acids? Nucleic acids mean DNA and RNA. So here in this case, you can say the Golgi bodies that don't contain mitochondria and chloroplast, both they contain DNA or RNA. So D is the correct option in this case. So students, now I am moving towards the question number 6. And question number 6 is, some features of the cells are listed: cell wall, cell surface membrane, and ribosomes. These are three cell structures. Which features can be found in plant cells and in prokaryotic cells? Plant cells and in prokaryotic cells. Cell wall is also present. Cell surface membrane is also present, and ribosomes are also present. So in this way, we can say the A is the correct option. Question number 7, which bonds are present in all viruses? In all viruses, you know, nucleic acid, DNA or RNA is present, and protein is present. So protein coat is present. So phosphodiester bond are present in DNA and RNA. Peptide bonds are present in proteins, and covalent bonds are also present. In this case, all three types of bonds are present in all viruses. So question number 7 has the correct option A. So students, you know, they are different types of bonds. Now, question number 8. A student carried out the Benedict test on 4 different concentration of glucose solutions, then recorded the time taken for the first appearance of a color change, the endpoint. The student found it difficult to identify the first appearance of color change and consistently timed each solution for 2 seconds. Each solution for 2 seconds more time. After the color change first appeared, this introduced a source of error into the experiment. Definitely, it can be. Which statements about this error are correct? The effect the effect of the error will be reduced if the student performs 3 repeats at each concentration of glucose. It will take too much time. For you can say already there are 4 different concentrations, and 2 minute when 2 minute already he has added into the time. So when he will repeat, it will take too much time. The error will prevent the student from identifying which solution has the highest concentration of the glucose. So this is also the wrong statement. Now next is the error is systematic. Yes, this error is systematic as he has consistently timed each solution for 2 seconds after the color change first appeared. So actually source of error was arrived over there. So third statement is correct. So according to third statement, only D is the correct option here in this case. Question number 9. Which statements about amylopectin and glycogen are correct? Amylopectin and glycogen contain 1,4 glycosidic bonds. Yes, they are. They have. Amylopectin contains beta glucose. No, amylopectin does not contain beta glucose. Glucose is present in cellulose. So this is the wrong statement. Glycogen contains more 1,6 branches than amylopectin. This is also correct. So according to this, the statement number 1 and 3 are correct. So the answer is B. Question number 10. And what is question number 10? How many statements about fatty acids are correct? Fatty acids are either saturated or unsaturated. Yes. Fatty acids always have at least two double bonds. No, not all fatty acids have two double bonds. So this statement is not correct. Long chains of fatty acids are very good energy source stores. Yes. Fatty acids are insoluble in water and do not affect the water potential of the cell. This statement is also correct, you know, students. So only one statement is incorrect in this case. So C is the correct option. Now question number 11. And question number 11 is, the diagram represents an amino acid. And you can see amino acid R represent a variable site. This is the R. What is not a possible side chain? What is not a possible side chain? You can say the chains CH3, second chain B, third, and this is the longest chain, which cannot be a side chain. So it means D is the correct option in this case. Question number 12. Question number 12 is, question number 12 is, the graph shows the effect of an enzyme on a reaction. And here in this case, you can have two chains, energy of state, substrate. So it is here, it starts from here, and then you can see it is very rate of energy of the system where, and then this is low energy use. So here X is can be the uncatalyzed reaction. So it is X is uncatalyzed reaction and Y is catalyzed reaction. And this is the Z is the amount of overall amount of energy released during this reaction. So according to this information, D is the correct option. Question number 13. Moving towards question number 13. The question number 13 is, the graph shows the results of two experiments on the effect of increasing substrate concentration on the rate of an enzyme catalyst reactions. One experiment was at a high concentration of enzyme, and the second was at the low concentration of enzyme, this one. So what would limit the initial rate of reaction? And then substrate concentration is on one side. What would limit the initial rate of reaction at point X? At point X, initial rate of reaction. So it means when there is high concentration of enzymes, it means more active sites are available. When there is low concentration of enzyme, then the you can say less active sites are available at that time. So what would limit the initial rate of reaction at this point X? When, and what he has done, only at this point when the substrate concentration, he has increased the substrate concentration. So when there will be the at point S, we are talking about the lower graph. So it shows that when there are low concentration of enzyme, substrate will be in high concentration. So there will be the less availability of active sites. So at this point, the enzyme concentration will act as a limiting factor at point X. So A is the correct option in this case. Question number 14. Which statements about the Michaelis-Menten constant of an enzyme are correct? All the Km values, half the active sites, half the active sites of the enzymes should be occupied by the substrate. Yes, this is correct. You know, this the half of the Km. So this is correct. Km represents the substrate concentration at which the enzyme is working at half its maximum rate. Yes, this statement is also correct. Lower the Km value, the lower the affinity of the enzyme to its substrate. No. If lower the Km value, it means the highest the affinity of the enzyme to its substrate. So this statement is not correct. When an enzyme has a high Km value, the enzyme catalase reactions will proceed very slowly to its maximum. This statement is also correct. So B is the correct option. First, 1, 2, and 4, these statements are correct, and B is the correct option. Which component of cell surface membrane helps to reduce fluidity of the phospholipids at high temperatures? Phosphate group, no. Unsaturated fatty, no. Proteins, no. Only cholesterol is that substance which helps to reduce the fluidity of the phospholipids at high temperature. So C is the correct option because it is helpful in controlling the fluidity of the cell membrane. I would like to move towards the next question number 16. So question number 16 is here. You can see over here. Plant cells were left for 50 minutes in different sugar solutions, 10%, 5%, and 1%. The water potential in the cytoplasm of the three cells was the same at the start of the experiment. The diagrams show the appearance of the cells after 50 minutes using a light microscope. And you can see in first of all cell P, it looks okay. Vacuole is not very large, but in case of cell Q, it is looking very turgid as it has absorbed lot of water, and cell R is looking plasmolized. So it means if it is plasmolide, it means it was kept in 10% solution, it was 5%, and it was kept in 1% solution. So what conclusions is correct? Cell P has the same concentration of sugar inside the and outside the cell. No. Cell Q is flaccid and cell P is plasmolized. Cell P is not plasmolide and cell Q is not flaccid. So this statement is again not correct. Cell Q was placed in 1% solution. Yes, it is looking turgid, you know. The sugar solution outside cell R. Outside cell R has a less negative water potential. Less negative water potential means it can be in negative. So if it is a negative, it means the water has more water molecules into it, due to which this statement is also wrong. Only in this case, the C is the correct option. Question number 17. We are moving towards 17 question. The graph shows the effect of increasing the side length of agar cubes on the surface area and the volume of the cubes. Surface area or volume. Here you can see this surface area and this is volume and side length. Which row correctly identifies line 1 and line 2 and the effect of increasing side length on the surface area volume ratio? Effect of increasing side length on the surface area volume ratio of the cubes. And in this case, you can see that if surface area volume ratio should be, you can say the difference between them, the cube should be of smaller size. It means their surface area will be high. So here in this case, you can say the one will represent the volume, will represent the volume, and number two will show the surface area, and third decrease or increase. So here, if volume, surface area to volume ratio will be decreased if there will be increase in the side length. If there is increase in the side length, then surface area to volume ratio will decrease. So according to this, C is the correct option in question number 17. Question number 18. Question number 18 is, a human embryo consists of 12 cells at the start of day 1. 12 cells. Day 1. The cell divides by mitosis for 4 complete days at a rate of 1 division every 32 hours. 4 days. 4 days mean 24 into 4 is equal to 96 days. And complete days at a rate of 1 division every 32 hours. So we will divide 96 by 32. So it will be 3. So it means 3 divisions will be there. So at the start, there were 12 cells. In the second division, they were 24. In the third division, they were 48. And in the third division, it was 96. That is, they were 12 in the first division, 24 in the second division, 48, and in the third division, they were 96. So according to this, the question 18 has the correct option B. Students, you can calculate by this way. Next question is question number 4. And question number 4 is, which diagram shows roles of mitosis? Roles of mitosis in number 1, you can say type of asexual reproduction and sexual reproduction and growth and regeneration. So in this way, all of these 4 types of options have one thing common, that mitosis is taking place in them. As you know, the mitosis is actually involved in the growth, in the cell repair, tissue repair. So it means all these 4 types of diagrams have one thing common, that is mitosis is taking place in them. So A is the correct option in this case. Now, question number 20, and question number 20 is, which events are part of the mitotic cell cycle? Which events? Which events are part? Events means mitotic cell cycle. They are talking about mitotic cell cycle, not only the mitosis. As in the previous question, they were talking about the mitosis, but here they are cell cycle. So interphase, anaphase, cytokinase, all they are involved, all they are present in mitotic cell cycle. So A is the correct option in this case. Now students, thank you so much. Please like, share and subscribe my channel. Thank you so much.